introduction. My name is Dr. Raquel Pleskis. I am a National Science Foundation postdoctoral fellow at the University of Connecticut in the Department of Anthropology. I'm really excited to be here today to share with you guys the results of the ancient DNA research of the archaeological individuals found at Nassau Street. And I also want to start by saying thank you to all of you, to the descendants, for trusting me to take on this research project. It is an honor and a privilege, and it's very, very serious work. So I hope that this information can help contribute to information that we know about these individuals, about who they are in their life, and what we know about the individuals that attended First Baptist Church. So I'm also going to be showing some images of some of the skeletal remains that were used for DNA testing. And we're showing these images to sort of illustrate to you guys how much sample was taken, what happens in the process of doing the ancient DNA testing, so mostly for educational purposes. So I also ask the same to not take any photographs when we're showing any of these human remains. Okay. Oh, of course.
We're in the process of trying to mute all. We have several people on Zoom that want to have their mics on, so we're trying to do that.
If anybody on Zoom has their mic on, can you please mute it? Okay. And I'm going to try to talk into the computer and talk to folks here. So, all right. So, what is ancient DNA? Well, ancient DNA is really just, again, code for just really fragmented DNA. It's just DNA that's not very nicely preserved, and that's because it's found in archaeological materials, organic materials that have been degrading over a process of 50 to hundreds of years. So for First Baptist Church, the DNA sources that we used were actually human skeletal materials. So if you see here in this top corner, let's see if I can get my little pointer. We used petrous temporal bones. So if you see the picture of the human skull, there's a little bit of a red area that's highlighted. That's the area which contains the petrous. So the petrous bone is bone that surrounds the inner ear. It's very, very dense bone, and so the denser the bone, the better the DNA preservation. So we typically try to use petrous, or sometimes teeth are also really good sources. So we use and obtain and extract the ancient DNA to try to answer three kind of main things. The first one is ancestry or genomic similarity, so how similar their DNA is to other contemporary populations across the globe. We can look at chromosomal sex, so X and Y chromosomes, to determine if the individual is a chromosomal male or female. And we can also look at biological kinship. If there are multiple individuals which we are able to successfully extract DNA from, we can see if they're related to each other. Okay. So with the First Baptist Project, we set out to really think about four main questions. So what is the ancestry or genomic similarity of these archaeological individuals? Are they of primarily African descent? What is the chromosomal sex of these individuals? Do we have any males here? Are they female? Are any of these individuals related to each other? And then what are the prospects of genetically connecting with living relatives today? Looks like animations might be a little bit off on this. All right. So as Jack pointed out, three individuals were excavated. We have burial 1, burial 13, and burial 26. So 2 bone samples per burial were selected in order to maximize chances for DNA recovery. So if one sample doesn't work, then we can try to use the other one and see if we get better results. For burial 1, we selected cranial fragments, given that the remains of that individual were pretty poorly preserved, so this was kind of the best that was available. Burial 13, we selected both right and left temporal bones. So again, thinking about that graphic on that first slide, that area near the inner ear, which is really good for DNA preservation. And burial 26, we selected the same. So those bone samples were then transported to the University of Connecticut in September 2022. So you can see here myself and then Jack Gary are beginning the process of cleaning some of these bone samples at UConn. So the first part of this process to do ancient DNA first begins with cleaning the bone microbial DNA that could be on it, or potentially any DNA from any people who are handling that. So this is the image of burial 1 post-treatment. So we'll do a bleach treatment. We'll wipe down the bone with a 50% bleach solution and then rinse it 3 times with water. So WB1 was very fragile. So if you pushed on it a little bit too hard, it would start to begin to crumble. Burial 13 really had nice intact bone, but then when we applied the bleach treatment to it, it really began to degrade and kind of just begin to crumble. So the integrity of the bone was not very good. And burial 26 was the most robust of the 3 individuals. And you can see that you have this little nice white piece of bone here, and that inner petrous portion. That little hole is kind of where your ear canal will begin. So very nice and robust and firm. And I'd like to talk about the states of these osteological preservations before we get into the DNA results, because they largely are connected. When you have poor osteological preservation, the DNA often is not very well preserved either. So after that, then we move to grind the sample into a small fine powder. Burial 1 and 13, because they didn't have a ton of integrity to the bones, we manually ground that with a mortar and pestle. Burial 26, because it was very, very robust, we do an automated sample grinding using something called a mixer mill, which will take that kind of metal ball and put it into the kind of the metal casing, and then that ball will go back and forth very, very quickly, and that actually creates that bone powder. So you can see there's that bone powder, which will then be used for DNA extraction. So this is the image here for another project, but it kind of helps you show what this process looks like. The bone powder is at the bottom of the sample tube, and then you kind of have this yellow solution. That is that DNA being kind of leached from that bone into that solution. So the process of DNA extraction starts by taking your bone powder, adding an incubation solution, that DNA goes into the solution, and then we will take that solution and pass it through something called a silica membrane. So you can see there's all the little DNA fragments kind of go into the middle of that membrane. They get stuck there. We wash it, we clean it, we remove all proteins and salts and other things that we don't need until that DNA is nice and cleaned, and then it is released. So basically it's just one long process of isolation. Okay? So now keep in mind that DNA extraction protocols don't selectively pick human DNA. They'll select all DNA that's found in that. So that can be things like our ancient human DNA, it can be human DNA from living folks today, or it can also be microbial DNA. It's going to pick all the DNA out. So you want to keep this in mind as we move forward. So we check at each part of the step to make sure it was successful and that there is DNA present. We do this through different quantification measures. So for burial 1, we found that there was DNA present, however, it was very little, so small amounts of DNA. And burials 13 and 26 all had DNA present, so we were all good to go. So this, to me, was a really great sign, because sometimes you don't even get DNA successfully extracted at this first step. And as well, our negative control was blank, which means that there was no contamination in the sample extraction process. The next step is then to do a library preparation. So what that means is basically we have all of our DNAs, and we want to add little tags to the end of those. So these tags serve 2 purposes. The first one is that they are able to differentiate between the different samples. So it's kind of like a Dewey Decimal System at the library. You have a bunch of books and you need to label them, you give them a code. Same thing with the DNA. This code also allows it to be read by a sequencing machine. So we need this to be able to kind of process it down the line. So we will add these little tags or adapters to our DNAs, and then we create thousands and thousands of copies of that. So it allows us to take something that is so small and make millions of copies, so we're able to better read them for sequencing. However, when we do this, if there's not enough DNA present, then we can't do the library preparation step. So here, burial 1 ended up dropping out. There just wasn't enough DNA in the extract for us to do successful library preparation. However, burial 13 was successful, burial 26 was also successful. Our negatives were blank and our positive control worked, which means that the protocol was a success. So we left burial 1 at this step, and we moved forward to our last step, which is enrichment. So you remember how I talked about in DNA extraction, we'll extract things like microbial DNA and your human DNA. Well, this step will basically allow us to pull out only the DNA that we want, so only the DNA for this archaeological human individual. So we do something called a hybridization capture or a whole genome enrichment, which basically will go through pull them out using this very cool magnetic beads, and this allows us to increase our DNA sequencing efficiency. However, we're targeting only for human DNA. So if your DNA sample is full of microbial DNA, that sample is not going to work. And so this is what we found happened with Boreal 13. This is our quantification. You can see there's this chart which shows the station result. We have basically 3 different peaks. The first and the last is a ladder, so ignore those. That's just how we kind of can quantify. But the middle is what we want to look for. You can see Boreal 13 does not have that nice DNA hump, meaning that there's no human DNA present, but Boreal 26 has a really nice hump. So we ended up moving forward with only sequencing Boreal 26, okay? So we submitted it for sequencing about a month ago. It was done on an Illumina HiSeq machine, which does next generation sequencing. We obtained a lot of sequencing reads you can see here in our table. However, about only 3% of those reads match the human genome after quality filtering. So this may seem very low, however, this is very normal for ancient DNA. So again, remember how I'm saying ancient DNA is just really fragmented DNA. It's not great DNA to work with. Well, this is a very characteristic result. Our coverage is about 0.73x. And so what I mean coverage is that we get a lot of different sequencing reads and we have to overlap them to kind of create what is called the consensus sequence. So if you have low coverage, you just have less data, right? Less overlapping reads. So it's a little bit harder to kind of make precise estimates. High coverage is something where you have a ton of data. So we have less than 1x coverage, so this is considered low coverage. However, again, very normal for ancient DNA, and so we're able to move forward with our next analyses. Okay, so this is where we get into the results after we've gone through all of this process, okay? So again, the first part of doing an ancient DNA analysis is we have to authenticate our DNA. You saw in the pictures from Jack Derry that folks were wearing Tyvek suits during the sampling, and that's really to make sure that we're not contaminating that DNA from all the archaeologists, because contamination will really just eat up all of our sequencing reads. We need to make sure that our DNA is authentic. So one of the big things that we look for are signs of damage. DNA degrades over time, and we see very specific patterns of damage at the ends of sequencing reads. So they just have a lot of damage. So that's kind of the signal of what we're looking for. So this is a map damage program, and you can see there's little upticks on each side, or kind of have a little bit of a smiley face. So for ancient DNA, that means good. We have a little damage at the ends of our sequencing reads, so we can move forward and say this is authentic. The second is to assess for contamination, like I talked about previously. We have nice contaminant DNA. That is primo for sequencing machines. That's going to want to sequence way easier than an ancient DNA. So we really want to make sure that we have a nice clean sample. We checked this using mitochondrial DNA program called HaploCheck, which indicated that we had no contamination. So thank you to all the archaeologists and everybody who worked on this. You guys, it really, really paid off. So thank you all so much. Okay, so now we can kind of get into the meat of this all. So our first step was then to estimate the chromosomal sex of this individual by basically analyzing it and seeing if there's any Y chromosome DNA fragments found. So we run a program by Skoglund et al., which basically measures the proportion of X and Y mapped reads. And we did, in fact, identify Y chromosomal DNA fragments, indicating that this individual was likely male. Next, we looked at whole genome ancestry estimation. So what this does is we basically look at there are 23 chromosomes. This is what we call our biparental ancestry. So you get your DNA from your mom and your dad, your biological parents. They get their DNA from their biological parents, and so on and so forth. So your whole genome represents kind of your total ancestry, okay? So we will rerun a principal component analysis against markers on what is called a human origins panel, which is a global reference panel. It looks at approximately 500,000 markers in your genome. And then we plot it on something called a principal component analysis. So when we're looking at this chart, we want to see each dot represents one person, and each color represents a most likely this individual is of African descent. Next, we do something called an admixture analysis. So admixture basically estimates the amount of ancestry clusters this individual has to basically see if, is it this individual mostly of African ancestry, or do we have European ancestry, Asian ancestry, Native American ancestry also present. So burial 26 largely has this big red component. So how you want to read this plot is basically each bar represents one person, okay? And then the different colors represent different ancestry components. So you can see you have a lot of big red in burial 26. Well, where else do we see a big red chunk? Also in sub-Saharan populations. So this indicates this individual is largely of African descent. You also may notice there's a little bit of yellow and green. However, it's pretty small, and that I think is mostly just kind of a random noise given that we have lower coverage of this individual. If the proportion of those colors was much bigger, then I would say that maybe there's some differential ancestry components. So again, we're thinking of African descent. Because we have a Y chromosome here, we can also do Y chromosome haplogroups. So we characterize this individual to have a R1b1a haplogroup. And this is actually a super interesting result because R1b1a has a very wide distribution. It's actually majorly found in populations of European descent. However, there is one lineage through which this individual is very closely affiliated with, which is in sub-Saharan and Central Africa. So we need to do slightly more comparative estimates to make sure that we kind of know what lineage specifically this individual is, but likely, given the whole genome estimations of ancestry that we just read over, it's likely an African lineage. And last, we did our mitochondrial DNA. So our mitochondrial DNA is the DNA of our mothers, right? You obtain your mitochondrial DNA from your mom, my mom gets it from her mom, so it allows us to say our direct maternal history. So for this individual, we characterize it as an L3D1 haplogroup. L3D haplogroups, this is from a paper published about 10 years ago. It's found broadly across Central Africa. It's not a super frequent haplogroup, however, we do see it also in African American populations today. So again, this information is largely indicating this individual is male and of African descent. So kind of in summary, we have 1 of 3 individuals who were successful in generating ancient DNA profiles. This was largely due to the state of osteological preservation. It just was pretty poorly preserved. So if the osteology is poorly preserved, usually the DNA is poorly preserved as well. Again, burial 26 is likely a male of African descent. And coming back down here to give this presentation, we've also packaged up all the remnants of DNA samples. So at each part of the process, we generate different tubes of DNA. So those are all been packed up and returned to Colonial Williamsburg for their eventual reburial. So we have no more DNA sample at UConn. So it's always a little bit of an emotional time to pack everything up. It's kind of the end of the analysis. But again, I just thank you all for your trust and letting me take this work out and for letting me report back these results to you. And so in thinking about future testing, about genomic connections to contemporary communities, when we think about our autosomal or whole genome DNA, now that we know that these individuals are associated with the first church, they're likely slightly too old to be able to do any autosomal estimations of kinship. So as we go back every generation, we lose 50% of our DNA. So if we're thinking this individual is from the first half of the 19th century, we're looking at like maybe 9 generations back, 7, 8, or 9 generations back. And so given that we have a lower coverage with this archaeological individual, and we already have maybe only about 2% of DNA that would be in common, the probabilities are very, very low, so it would not be possible. Y chromosome and mitochondrial DNA lineages could be a possibility. However, these lineages don't provide direct assessments of relatedness. So you and me could share mitochondrial DNA, and we may have the same mother or grandmother, or we could share an ancestor 2,000 years ago. So it's not able to definitively say any kind of relatedness. With this, we're going to really need to be able to do lots of really great genealogical research and being able to actually identify who the burial is itself. So with that, I'm going to turn it over to Dr. Joe Jones.